Hello everyone. In this tutorial we are going to talk about primer design, and we are also going to look at how to design primers from a sequence generated or obtained from Genbank. How do we select or identify our coding region? How do we select primers? What are the principles behind it? And we are also going to do some calculation on how to estimate your fragment size and as well as your protein size of the selected region. My assumption is that you probably watched the PCR video, and this is just a continuation of it. Primers are short single-stranded DNA fragment that anneals to the region upstream and downstream of your amplified target. So I'm just going to do another drawing here. If this is your DNA region, this entire strand here, like this, and this as well. Now, what you want to do from our PCR reaction, you want to amplify this region that overlaps with the red. Okay. So if this is a region that you want to amplify, or you want to have more copies of for a specific region, what you need in a PCR reaction, you need primers that are going to anneal or hybridize here upstream and here downstream of your target region. And the reason why they anneal here, they're going to make it easy for polymerase to later on bind here and start the synthesis of this region a couple of times, and at the end of it you are going to have multiple regions like this of those red regions, and the blue are actually eventually going to fall away. So that's the reason why we have primers. And it's important to understand how do we create the primers and what are the properties or what are the characteristics that we must consider when we want to design primers. The first one that we're going to talk about is the length of the primer. How long should your primer be? The second one is your annealing and melting temperature. How do you adjust them and how do you go about them? The third one is the GC content, and the last one is going to be the secondary structure of your primers. So without further ado, let's get straight into it. Now, when it comes to the length of your primers, what you should consider is the primers that are too short. For instance, if I still show you a region here like this, primers that are too short, they are going to have very region that are non-specific. For instance, if I just call out, you have a sequence and you only have 4 base pair of A, G, T, C, or let's say 5 base pair like this. This is too short because there's a high probability that you're going to find region like this in a larger piece of DNA, and too long as well, it may result in hybridization break. So the ideal, this is of course most people would argue 15 or 20, but this is quite the acceptable range between 18 and 25 base pair. This is actually ideal and it should give you the best yield. Next is the annealing and melting temperature. So annealing temperature is what allows the primers to base pair with a DNA. So at this point, your annealing temperature is what allows this primer to actually base pair with this region like this. All right. And your melting temperature is actually the temperature at which half of the primers separate from the DNA. So they separate to make room for the next reaction to occur and so forth. Now, how do you go about it? The ideal is that your annealing temperature should be about 5 degrees Celsius below the melting temperature. At least in that way, most of the primer will bind to the template. And also, please do not forget that when you choose this temperature for your forward and reverse, you must keep them the same. So if you have a melting temperature of, let's say, 55 degrees Celsius, then your annealing temperature should be around 50 degrees Celsius. Of course, this is not written in stone, and this change may vary depending on what region you're amplifying, but at least from this point of view. There are exceptions, but for us to understand the basics, that's something that we should take into consideration. Next is your GNC content. What we know by now is that between our base pay, like this, what we have is that there is quite a difference between the hydrogen bonds. Some of your bases, like the ANTs, they only have two, while the GNCs, they have three hydrogen bonds. What does that tell you? That when we need to break those bonds, the ENT will be easier to break than the GNC. So the ideal composition here is that you should have a GC content or percentage that ranges between 40 and 60%. And what is important as well, you can include a GC clamp. So about a couple of base pair like this of GNC at the end of the primer, just to make it more stable. Because if you have anything below 40%, which is going to be very rich in ANT, and it's going to provide you weaker bonds, easier to break. And if you have something above 60% as well, that is most probably going to be as well, too difficult to break, and that's not good. And last but not least, the secondary structure. So when it comes to the secondary structure of your primer, what we're talking about is the formation of hairpin-like structure like this, where your primer may actually fold on itself, or the primer will anneal with itself without actually annealing with the piece of DNA. If these are your primers, they may form hairpin structure like this, or anneal with itself without actually binding the region. So they form hairpins, self-dimers, or cross dimers as well. So that's something that you should really look out for. But in practical, you could have tools that may help you predict that, and also avoid a lot of repeat sequences, because they may lead to this as well. So what have we learned? That length of the primer between 18 to 25 base pair, not too short, not too long. With your annealing and melting temperature, this should be within a 5 degrees Celsius range. Your GC content 40 to 60%, and the secondary structure of your primer as well. You need to avoid repeats and try to use tools that are going to predict the presence of cross dimers and self dimers as well. Now that we have this information, let's dive straight into it. What I have in front of me here, what we are looking at is mRNA fragment, or a nucleotide fragment, and what we'll try to do here is to design the primer. Okay, let me just use a nicer color. We will design the primer, but before we design the primer, what we need to find is actually the coding region. And this coding region, we refer to it as a CDS. And from this coding region, we are going to also try to get the size of our fragment, and after that, we're also going to know the size of the protein product. All right. So when you're presented with a Genbank page like this of a protein, for instance, this is a lacase from a polyporous brumalis, which is probably a fungi. Yes, we have a fungi there. You look out for the size. So this is the size of your fragment, and you also pay attention to this, the CDS. So it's telling you that in this nucleotide, this entire sequence here, as you see, all of this, you have about 1,804 base pair. And in this 1,804 base pair, if this is your full 1,804 base pair, your coding region, or your region of interest, that as a start and probably a stop codon, is going to be from position 78 to 1652. All right. So what is the actual size of your fragment then? In order for you to work that out, all you need to do is to perform 1652. You can take up your calculator, minus 78. So the size of my fragment of my CDS, So my CDS size then is going to be 1652 minus 78. Since this is inclusive, then you're going to have 1754. We also want to include this first region here, the first number there, then it's going to be plus 1. You add a 1 there, then it's going to give you 1575. Then that is the size of your coding sequence. Then once you have the size, we may also assume that a protein, let's say protein is 100 DA here, then what you want to do to get your protein, all you need to do is actually subtract. So 1575 minus 3 because of our codon, the stop codon. You remove the stop codon here. What you are going to end up with is 1575 first minus 3. You have 1572. Then you are going to divide this by 3 to actually get the number of amino acid present. And once I divide it by 3, I have 524 amino acid. And since here, let me just clean it. You feel free to pause and rewatch the video as many times as you want to. I'm just going to clean here now that we know what's happening here. We going back to the assumption that one amino acid is equal to 100 DA. Then you do this number times 100 DA, which is going to give you 524 DA like this. So that could be the potential size of your protein. So once you have all this information, which is fine, we're going to remove that again, and we're going to start on a cleaner note, and now start designing our primers. What have we said? Just a quick reminder that our coding region here is between position 78, so we need to find position 78 here, and it stops at 1652. All right, so how do we go about this? There are many ways in which you can do this. I'm just going to remove maybe this one for now. We don't really need it. All you have to do is to look for the coding region. Here, the nice thing is that we have this base pair or this base pair in bunch of tens. Okay, so this is 1, 2, 3, 4, 5, 6. So this is 60. So since we are after position 78, what we're going to assume, this is 60 and this is 70. And if you count 1, 2, 3, 4, 5, 6, 7, 8, you should land on this ATG. So the first A that you see here, this is actually position 78, and I just took it as a full codon because I want to make sure that that is actually a region that code for something. And if I remember my genetic code, what we know is that a codon is made of 3 nucleotides, and your start codon is always methionine, which is AUG. So if I take my ATG region here, what I do a complementary base pair for it, and I get an mRNA strand out of this. It's going to have A, U, and G, which gives me now the confidence that this makes sense, that this A here would be position 78. So that's how we get this. And the end of our sequence is in position 1652. You do the same, so you end up here 1620, 30, 40, 50, 60, 61, 62. So this T here is actually where we end things. That's where my coding sequence is. So if I were to highlight my region, I'm going to select like this. Of course, there are nicer ways of highlighting the whole thing, but at least you get where this is going. So this region here is my coding region. So that's how I found it. So 16, 20, 30, 40, 50, 20, 30, 40, 50, 60, and 62 there. So that's how we actually identify this. And feel free to watch this over and over and over again if you are still not sure how we got this. That's the reason why we have the video. You can slow and count with me and just make sure that you understand it. So now that I have this region, I'm just going to clean it a little bit so we can carry on with our work. I will underline my AT region here, just like that. And I'm also going to lock it here just to emphasize that this is where we are working with. And that is now, maybe if I assign it a different color, maybe in this green, this is the region that we want to amplify. This region here, starting from this ATG, going down here up to this. So this is the region that we want to amplify. So if this is our very beginning sequence, which we said the size of the fragment was, I think, 1,804. So out of this 1,804, the region that I've highlighted in green here, let me just give it a green color as well. The region I've highlighted in green, okay, the green is a bit tricky. All right, so this is my CDS, my coding region, as we said, from 78 to 116.52. So I want a PCR that is going to create multiple of this region. Of course, if I want that, I would have to design primers, and those primers would have to anneal to the beginning here and the downstream part here as well. How do I go about it? There are many ways and many factors that will allow you to select. Sometimes, you may want to select region that are a little bit further from this region and maybe a little bit down here, just to make sure that you incorporate everything. That is also okay. But for this tutorial, just to make everything easier, I would like us to just select from this and going down as well, just for uniformity, so that we can all follow a single thing. But in real life, remember we spoke about the length of the primer, the GC content, annealing and melting temperature. You may see that you may want to go a little bit back before your actual start or after, so you can incorporate and have the optimum condition. So how do we go about it? We said that the first thing that we need to pay attention to is the length of your primer. So talking about the length of the primer, I would just make a decision for all of us in this tutorial to go with about 20 base pairs, and 20 base pair, of course, upstream and downstream. I've already done it for us, and you can also do it in your own time. My first region here, if I remove this and I create a clean one, I've selected here from my ATG. I've selected about 20 nucleotide, which is this full region. So I went with it like this. I'm going to start here for my 20 region. And for this one as well, I think we're somewhere at the bottom here. I also selected something like this. And there we go. Now I know that my primers, they should anneal to this sequence and this sequence as well. So since I've already done that, if we look at the primers here, here's what I've done. what you would notice is that for my forward primers, since I selected the sequence, ideally what I would have to do now is to provide a complementary of this, which is actually going to be my actual primer. Then I'm going to have T, A, C, A, G, and so forth. You get the picture. So my actual primers are going to be this one. This is just this region here that I brought here. I hope that that makes sense to you. And all you have to do is just the complementary of this region. That is going to be your actual primer. And you get the picture. It's very straightforward. It's going to move in this direction. For your reverse region, which goes in this direction, this is a little bit tricky. But again, it's not necessarily too difficult, or like the end of the world. You should be able to still get something good out of it. So what have I done for this specific region? For these reverse primers, of course, I selected my region. I think what I did here is I went a little bit back up to here. Let me just show you the sequence that I actually went for. So we have a couple of CCC. So I actually took this entire region here, which is technically a little bit way more than what I was supposed to take. But I'm just showing you as an example. So please make sure that when you do select yours, you actually get until the end there. So as you can see, I think 4 cytosine. These are the 4 cytosine, and I stopped. Let's look at it again quickly. 4 cytosine, and I actually stopped somewhere before there. So we're going to reselect that again. 4 cytosine, and we have a region up to ACT. Okay, so this is what I selected, but in fact I was supposed to go up to this point again, but it's fine. What I'm going to do, we can just redo that quickly. I'm going to take, while we are actually on it here, I'm going to take that region quickly and show you how we do it. I'm going to remove this one, and maybe let me keep this one as well, like this. So what we have here, I'm going to rewrite my forward first, which is now T, A, C, T, G, C, T, G, T, A, A, G, G, and we have T, C, T, and you continue. I think hopefully you get the picture here. So with this bottom part, let me select the right sequence. So I'm going to get from this region here up to this one, and hopefully these are the 20 sequence. So if you just give me a moment there, I'm going to select that for us. And once we have the region, then we are going to actually get our DNA strand. So we are just selecting it now, and I'm going to show you how it happens once we have the region. We are going to create a complementary base pair of it, which you're not going to see now at the moment, but once I'm done with it here. In fact, let me just show you. Actually, this is where I'm selecting it from. Once I have it, I just wanna remove all the different colors and all that. I'm going to select the region again, and I'm going to work out the reverse complementary of it, and I will explain what has been done with that reverse complementary region to make things quicker. I'm just using a tool, and we have the reverse, and we've also flipped it. All right, so here's what has happened. If I were to select this now and bring it back to us, you will notice that we actually flipped our sequence, but not only we flipped it, we also worked out the complement for it. Now let's have a look of what is going on here. Here's what was done, or what we just did from this region. So I selected this region. The first thing that I did is to get the complement of this. So this T became A, and this is now G, and this is C, and this is going to be A. And once I got this, I got to flip it now in such a way that I'm going to start with A, G, C, A. That's why you see here we have A, we have G, we have C, and we have A, and we continue so forth. And this is what is going to give us our 5' to 3' region for this bottom section here. So feel free to stop this video, watch it over and over and over again, just to make sure that you understand what I've done here. If I were to remove everything, I'm going to show you again one more time. The region that you see here, this is it here. What I did, I first found the complementary of it at this point here. I'm just going to reread it. This is now A, this is G, this is C, and this is G. Again, this is T, and this is what you got here: A, G, C, A, G, C. Yeah, I forgot an A somewhere in the middle here, but hopefully you get the picture. And again, if you don't mind, I'm gonna do it one more time for someone who did not get what was done here. Please look at my region again. I'm gonna use maybe the green. So this region is exactly what you got here. I just extracted it. If you want to do it quicker, I used a tool because I have access to a computer. But if you don't have access to a computer, all you need to do is just rewrite it. First find the complement and flip the whole reaction. And I'm going to redo it here like this. And this is going to become my A. And this is now going to be G, and this is going to be C, and now I got that A here. Then I have my G, and I have another T, and I'm going to stop maybe here when I have another C. So if you see, it's A, G, C, A, G, T, C, which is also here A, G, C, A, G, T, C. And that's how you actually find your 5' and your 3' there. I hope this was helpful, but feel free to stop the video, watch it over and over again until it all makes sense to you. All the best. Thank you.